Welcome to this platform. So in today's recording, we are going to revise transport in plants. So the topic we are revising is transport in plants. As always, we are going to answer exam questions. Now, at this point, please, if the recordings or the revision program has been helpful to you, you can just take a moment and of course subscribe to my YouTube channel, comment, invite some more of your friends also to benefit. Even as you prepare for your exams, I wish you distinctions. Okay, so this is the first question that we are going to answer. And this question is coming from 2016 school certificate. That was question 3. Okay, so here is the question. So figure 3.1 shows a cross section through a stem of a plant which has been previously dipped in red dye. So take note of this, which has been previously dipped in red dye for 6 hours. Now, before we even continue, let us identify what kind of a plant is this, or the one that has this kind of arrangement in terms of vascular bundles. And vascular bundles, I mean the xylem, the phloem, and of course the cambium, the way it is arranged. So this one is the dicot. Okay, dicot is a dicot stem. Okay, so this is the dicot stem. Okay, so this is the dicot stem. So that is something that you should know. Okay, or dicotyledon stem, the cross section. That is very important to know. Then now let us look at the questions. Identify the parts labeled N, P, and Q. So you are going to identify the parts labeled P, N, and Q. What are the parts? Okay, so in identifying the parts, let us look at N, P, and of course Q. What is our N? So N, as you can see, this is the line that separates the two parts, which is P and Q. Okay, so this is called the cambium. Okay, so this is the cambium layer. Okay, so that is the cambium. Then what about P? So P, the one that is on the outside part, is called we are looking at P. So this part P is the phloem. Okay, so the one that is on the outside is the phloem. Then of course the one that is on the inside, which is Q, is the xylem. So that is how you can look at them. So the one inside is called the xylem, and of course the one on the outside is called phloem vessel. So these are the parts we've identified them. So answer this question and 3 marks. Then which labeled part in figure 3.1 would be stained red? Which labeled part among these parts that we have, the 3, the cambium, the phloem, and xylem, which one of these will be stained red? So exclude the cambium. Let us go for phloem and xylem. Now it's very important for us to, as we answer this question, it's very important for us to know the function of each. This phloem transport sugars, okay, transport sugars and perhaps amino acids, okay, from where they are manufactured to the parts where they are needed. So the bottom line here is that the phloem transport manufactured food, but for the xylem it transport water. It transport water and mineral salts. Now look at this. It transport water and mineral salts. Now, if this ink that you are seeing, component of part of this ink is water. Okay, so now you know where it is going to move. It is not going to move in the phloem, but it's going to move in the xylem. And therefore here, the part that will be stained red is the inner part, which is part Q. So you can write part labeled Q will be stained red. And give a reason to your answer. So the reason is because we can say because part Q transport water and mineral salts. transport water and mineral salts, of which the dye is made of. So if the dye that we are seeing here, it contains some water, and therefore it will be moved along the xylem and not the phloem. So that is the reason that you can give. Then suggest the 3 factors that would increase the rate of movement. Now this rate of movement, we can look at it. This rate of movement, what are the 3 factors that would increase the rate of movement? Okay, what factors that would increase this one? So 1, temperature. That is the one of the factor. So this one we can just like look at more like the factors that affect transpiration. So temperature, you know that high temperature increase the rate at which water will be absorbed, and therefore the transporting part will be very high. Okay, so temperature or high temperature, you can say. Then of course we have humidity. Humidity, when you talk of, since we are saying that we increase, so humidity is the amount of water vapor in the atmosphere. High humidity, which means you have a lot of water in the atmosphere, and therefore for this one to move faster, there is need for low humidity, so that there's low amount of water in the atmosphere, and therefore more will be absorbed, and of course there's that concentration, there's that gradient that is created when there's low humidity, that results into increasing the suction force of absorbing the ink. Okay, then of course we can look at the wind speed, okay, or the speed of the wind. Now when the wind is high in speed, this process will be fast, so we can say high wind speed, one of the factors that would affect the increase. So these are the 3 factors that will affect the increase of the movement, or that would increase the rate of movement of the dye in the stem when it is dipped into the red dye. Name the process that would cause the movement of the red dye in the stem. Name the process that would cause the movement. So like I said already, these are the factors that affect also transpiration, if you have to look at transpiration. So get to look at also that part. That is another part that you need to consider looking at as you study this topic. That is transpiration. What are the forces that moves the water along the xylem? Okay. Then now, so here the answer is transpiration. That is a process that causes the movement of water, that causes the movement of the red dye in the stem. So the answer is transpiration. Okay, so we fully answered this first question. Like I said, please, on this part, so this is the dicotyledon stem, the cross section. You need to know also the monocot. How does it look? How is the arrangement of vascular bundles? And when we're talking of vascular bundles, we are simply looking at the xylem and the phloem. This makes up what we call the vascular bundles. And of course, as we go on, we might find questions what makes up the vascular bundle. So please look at that also. And it's very important also that you look at the external part of these. Aside from looking at the cross section of the stem, that is both for monocot and dicot, should be able also to look at the roots. So that is very important. Okay, so here is another question. Now for this question, please, I'll consider just putting the lines, how it is arranged here. So the lines, there are 3, I can put about 3 lines here, something like this. So now, the way it is here, the question is figure 4.0 shows the longitudinal section of a stem. Okay, so what you are seeing here is a longitudinal section of a stem which was just slightly cut. Okay, which later in figure 4.0 represent the cambium, the xylem, and the phloem. Now we are going to go back a bit. And as you can see, these are vascular bundle that you are able to see. These are vascular bundle, and looking at how they are arranged, it is not different from the question that we are from answering this question. So that is how they are arranged. As you can see, that one I pointed at, this is how they are arranged. So which means we can look at that as the dicot, okay, stem. So now the question is, you are going to identify those parts. Now just before we go there to identify them, look at this. This one is the cross section, but that one is cut on the side. So which means starting from outside, if we look at the layers, the one that is outside is the what? The phloem. Okay, the phloem. Then the one that is inside or in the middle, here, the layer that is in the middle is the what? The cambium, and of course the inside part is the xylem. So let us look at it. Let us look at the question. So what is the question? Which letters in figure 4.0 correspond to cambium? So since here is not visible, let's say there's a layer 1 and a layer 2 and of course layer 3. The layer 1 that is outside, let us say it is S. In this case, we are going to consider the layer S to be the one that is outside, and therefore we are going to say the layer S, okay, because here we don't know where it is, but we are going to say this is S, this is, what is the next one? T, and this is U. Let us put it like this, okay? Let us put it like this. Or let us say, okay, just like this. So which letters correspond to the cambium, xylem, and phloem? So please, the question might not have been like this, but after explaining, I'm sure you'll be able to get a thing. So which letter correspond to the cambium, xylem, and the phloem? So for a cambium, it is what? T. So T for cambium. Remember, the cambium is found between the xylem and the phloem, so right in the middle here. Then of course the one that corresponds to the xylem, it is U. So U equals to what? The xylem, okay, the inside part. Then of course S now is the what? The phloem. So I'm answering this question according to these that I've put here. Explain the function of S, T, and D, U. Now, since we've compromised the question, so we are going to explain the function of what? The phloem, and of course the what? The cambium. Okay? What is the function of the phloem and the cambium? Okay? So what is the function of the phloem? So from where we are coming from, we said the phloem, which is the outside part here, transport manufactured food. Okay? Transport manufactured food. That is what phloem, that is the function of the phloem. Then now, aside from that, it also transports amino acid, please. It also transports amino acids through the process called translocation. So that is very important to note. So it transports from the point of manufacturing or origin to the sink. That is the usage part or storage part. Okay, so that's very important. Then now, what about T, which is the cambium? What is the function of the cambium, which is T? So the function of the cambium, so for the phloem, we said transport manufactured food from the point of, from the source, let me say from the source to the sink. The sink is where they are used or stored. That is very important, but they also transport amino acid, please. Okay? So let us now look at the function of the cambium. So what is the function of the cambium? So the function of the cambium, which is T, cambium, let me just give the functions here. Okay? So 1, carry out cell division. Carry out cell division to produce new cells. The new cells that are produced include the xylem and the phloem. So that is the function of the what? The cambium. But aside from that, separates the xylem and the phloem, as we've seen in the previous one. So separates the xylem and the phloem. So these are the two functions of the cambium. So please, as we come across, even if we come across another question asking us about the cambium or the function of the cambium, we should be able to come back and refer to these answers. Then which letters correspond to the structure which need companion cells to function properly? So the structure that need companion cell is the phloem. So now we are going to write the letter. So which letter correspond to the structure which needs companion cell to function properly? So letter S, okay, according to what I've explained here. Then which letter in figure 4.0 correspond to a structure in which substance can either move up or down? So in the xylem, there's one movement or one direction in terms of movement of water. That is from the ground to the top. And of course, it is not in the xylem. And of course, the cambium does not do that. So it is the letter S, which is the phloem. So it is in the what? In the phloem, in the case you're asked to write the name. So that is basically what you needed to write. So this answers the question here. Let us see if you can look at another question. So this question came in a particular year as well. So this one came in 20. So the previous one that I'm just from answering, this one was in 2017. So be able to confirm 2017 school certificate. That is ECZ. Okay. Question 4. Then the next one, this one came in 2018 GCE. Okay. Now, let us look at the question. So, the figure 4.1 or 4.0 shows the vertical section of the vascular system in the dicotyledonous plant or stem. So, this is also a dicot, and we are told it's a stem. So now, and again, the key thing is that the vascular system is made up of the xylem vessels, and of course, phloem vessels. Now, where we are coming from, we raised or we answered a question, and it's very important to go back and look at the question, this question. Which letter represent the structure in which substance move either up or down? So, in the phloem, substance can move up or down. But since we are talking of the vascular system here, we know that one is a phloem, the other one is a xylem. And now let us see the question. Identify the structure T. Now, how are we going to identify the structure T? And I must admit that this question is also not clear, but I'm going to, by all means, explain so that you understand the thing. So now, identify structure T. So here in between there's a cambium, that separates the two. You know that. So there's a cambium here in between that separates the two. But there's one here and the other one here. Now, in this S, you see, the arrows are pointing up, which means the substance are just going in one direction, up here. And here they are going down. In the xylem, you never find things moving downward. And therefore, we can identify T as the what? The phloem. Okay? Identify structure T. So this is the phloem. Now, aside from moving the substance moving downward, right here at the end of it, there are cells here that you are not able to see. There are cells here. And these are what you call companion cells. Okay? So now, what two structures or features in the diagram that helped you to identify the structure or to identify T as a phloem? So we've identified as phloem. What made us to identify it as a phloem? So one, movement of, so one is the arrow. Let me put arrow. So the arrow showing the direction of movement of substance. So one arrow showing the direction of movement of substances. Okay, the direction of the arrows, where are they going? Downward. So that is one feature that helped us. Then aside from that, presence of what? Presence of companion what? cells. Only xylem, only phloem I mean, only phloem have companion cells, okay? Companion cells. And of course, in the way we are coming from, I'm sure we are going to find again the other question that is asking us about concerning companion cells, as we are going to see. Name two substances transported in S. What are the two substances transported in S? So S we said is the what? The xylem. Two substances transported in xylem, we said xylem transport water and what? Mineral salts. Very important. So water and what? Mineral salts. Now what about if it was in structure labeled T? What are the two substances? So one, sugar or glucose. Then also amino acid. So we might have answered the question that you might encounter as we go on. So take note of these answers. And of course the cambium, you know the function where we are coming from. Okay, I hope you've not forgotten. We said it carries out cell division and also separates the xylem and the phloem. That is question. Let us go to question C1. On the diagram, label the companion cells. So I've just done here. So the companion cells just needed to put an arrow here, not really circling them, but just to put an arrow. That is what you needed to do. So this one ends as a mark. Explain the role of the companion cell in the vascular bundle. What is the role of the companion cell? Okay? So what is the role of the companion cell? So listen very carefully here. So companion cell provide energy for active transport of substance. So provides energy for active transport of substances. What are the substances that are transported actively? Amino acids and glucose. Because these, they are transported up or down. Okay, so they need, or the cell need a lot of energy or needs energy. So that is the role of the companion cell in what? In the vascular bundles. That is very important. And of course, from cell specialization, if you had time to look at my video on cell specialization, I explained much on the companion cell. So if you have not done that, please get to look at the video on cell specialization where we answered questions. Then now, explain two functions of the cambium, chi. This one, we've already answered it. And of course, I'm just from explaining here. So carry out cell division. Okay, carry out cell division to produce new cells. What are the new cells that are produced? Xylem and also what? Phloem. Very important. Then now, what is the other one? The other one we said is separates xylem and what? Phloem. Okay, or separates xylem from the phloem. So that is the function. Okay, or these are the two functions. Okay, let us move on to look at another question now. So this question came in 2021, if I'm not mistaken, 2021 GCE. Okay. Then now, the figure 3 shows parts of the organ of a plant. So now, the first thing that you need to know is to identify which organ of the plant is this one coming from. So this is the internal structure of a leaf. So the organ is a leaf, in case we find the question. Then now, what are the questions? A, that is Roman numeral 1, identify the structure G and K. So what are the structure G and K? So now it's very important here to look at it very carefully, because there's another structure here that is the thin layer that is on top there. And this one I would label, it is called the cuticle. Okay? And it has a function. Okay? One of the function is to prevent that is excessive loss of water from the leaf. Okay, and it is shiny. One thing to know. Okay, what about G? This next part here, it is called the upper epidermis. So G is the upper epidermis. Upper epidermis. which means there's also a lower epidermis. So this is called upper epidermis. Okay. Then what about K? So K, where is K? So K is right here, and there's an opening here between. So the cells between here, the ones that cause, or the one that forms this opening, these are called guard cells. So that one is called guard cell. Now this opening, just to name it, is called the stomata. Okay, this is stomata. Then state one structure difference between structure G and D. So structure G, which is the epidermis, and J. So J is the whole of this one. So this one is the lower epidermis. So what is the difference between the upper epidermis and the lower epidermis? How many? So one. So one difference is that, look at this. This one, the lower part has what? The guard cells. Okay? It has the guard cells. That is one thing that you should understand. So structure J has stomata, while structure G has no stomata. So structure J has stomata, while structure G does not have stomata, or does not have. Okay, so let us answer question B. We are answering question B now. Identify the plant organ in figure 3. Identify the plant organ. What plant organ is this? I had said already, I said it's a leaf. So the answer here, the plant organ is the leaf. Okay, so the leaf is the answer. In which labeled region in figure 3 is the rate of photosynthesis the highest? So where is photosynthesis the highest? So photosynthesis is the highest in the region H. Okay, so photosynthesis is the highest in the region what? In the region H. Okay, and what is the reason why? So I can write region H. And what is the reason to why the photosynthesis is highest here? These cells that you are able to see, these are parasite cells, and they contain numerous chloroplasts, which traps light energy, and you know that is used during the process of photosynthesis. So how can you write? So you can say because cells in region H contain numerous chloroplasts. Okay, chloroplast, which traps abundantly light energy used to carry out photosynthesis. So, which traps a lot of light energy used to carry out the process of what? Photosynthesis. I hope you've gotten that point. So, because cells in region H contain numerous chloroplasts, which traps abundantly light energy, or a lot of light energy used to carry out the process of photosynthesis. Okay? These cells that are down here, although they contain chloroplasts, but they are not numerous. Okay? Few amount of chloroplasts there. Then now, let us look at this question. So I'm going to clean and of course just expose the question that follows. Then explain why the arrangement of cells in region labeled I is important for photosynthesis. Which one is region I? So this region I that you are able to see here. Look at how the cells are arranged. So they are scattered there, and one thing that you should note about the cells in terms of shape is that they are irregular in shape, and look at how they are scattered there. Now, the question is, why is the arrangement, the way they are arranged, important in the process of photosynthesis? You know that photosynthesis, for it to take place, it requires what? Carbon dioxide, and this carbon dioxide comes from the atmosphere, and it diffuses, it enters the cell through the what? Through the stomata here, okay, and this is able to diffuse through these spaces in the cells as it moves, making its way to these cells, okay. So in short, the reason why the arrangement of the cell or the explanation that we can give is that to create a large surface area for the diffusion of gases and, okay, for the diffusion of gases. So to create large surface area, create a large surface area for diffusion of gases, okay. I can also add and water molecules. It makes sense because here the gas that enters is the carbon dioxide. I think I've seen a question where they show the arrows, the one that is going in. So the one that is going in is carbon dioxide, and the process by which this one enters the cell is diffusion, please, in the case where you find such a question. So the gas going in is carbon dioxide, and of course through diffusion. And of course, after the photosynthesis has taken place, the gas that is produced is oxygen and it diffuses out. So what comes out is the oxygen, and also through diffusion. But remember, this opening, which is called the stomata, also is where water diffuses out of the leaf. That is the process of transpiration. So very important to look at, creates a large surface area for the diffusion of gases and also water molecules. And that answers the question fully. So this is what we had in 2018, in 2021 GCE. Let us look at perhaps the other question. So this is another question that we are going to answer. Now the question reads, figure 3 shows longitudinal section through a root tip. So this part, transport in plants and also development, that is growth and development in plants, that is where you find this kind of structures of question. That is where they are coming from. So it's very important for you to look at these two topics, that is transport in plants and of course growth and development in plants specifically. Okay, so now let us look at this question. Okay, so identify the parts labeled G and J. Look at the part labeled G. So G, these are the extension. Remember, from the statement, it shows the longitudinal section through a root tip. So this is the end part of the root at the tip. So at the tip of the root, you find this. Now, what is G? So G, root hair what? Root hair cell. Root hair cell. That is G. G is a root hair cell. And among the cell specialization or specialized cell, one of the specialized cell that you're going to find is a root hair cell. And its function is to absorb water from the ground. So absorption of water. Then what about J? So J is found right here. Now if you want to look at this root, it's at a tip. So at the tip of the root, we have the layer there that is down here that protect the root from mechanical damage, even as the root is penetrating the hard part or the rocks. So that part is called, or the one that shields the root is called the root cap. So this one is called what? Root cap. So it's very important to look at it as, the name is called the root cap. So root cap here. Okay. Then now the other question is, which labeled part in figure 3 is responsible for generating new cells? Now when you are looking at these, the parts, just to mention them or just look at them, this is a root hair cell. Now here in this region as on cell specialization, the cells are produced right in the region labeled I, and this is called apical meristem. Okay, apical, specifically root apical meristem. So meristem is just the growth center of in this case the root. So there's apical meristem, and also there's also in short, there's a root meristem and also the stem. Okay, so very important, apical, that is at the end. So this one, which is the question is, so this is the growth center, please. Growth center, which is called root apical meristem. That is the name. But the question is which region. So we are going to put region I. Okay, region I. That is where new cells are produced. And when they are produced, they move upward here as they start specializing, going up. Okay, you know the process of cell. It starts from division, then of course they attain vacuolation, then specialization. Okay, so in H, that is where vacuolation takes place in the case you are asked. Okay, then region H is responsible for cell differentiation. Okay, cell differentiation, and that is vacuolation. Apart from vascular bundles, state two other cells type differentiated by H. Okay, so apart from vascular bundles or vascular bundle, you know that vascular bundle we said contains the what? The xylem and the phloem. So in this case they're asking you, apart from the xylem and the phloem, which other cell? So other cells are called parenchyma or chorenchyma. Okay, so I'll write parenchyma. Then also we have the sledenchyma. So these are the other cells that you expect there. Then of course, state two types of cells found in the vascular bundle. So this question, I think I had echoed much, or I had echoed as we are where we're coming from. You might find such a question, and here is a question. State two types of cells found in the vascular bundle. So this one is simple. So phloem and what? Xylem. Then which named type of cell in C allows the movement of substance up and down depending on the needs of the plant? This one, we've looked at this and I had time to explain. So this one is the what? The phloem. Then suggest one meristematic structure not in the what? In the diagram responsible for bringing about secondary growth in the what? Dichotomous plant. So you need to look at also the types of growth. There's primary growth. Primary growth, we are just looking at the upward or the tonus, the increase in length of the plant. But if you look at the secondary growth, we are looking at the increase in the girth, okay, of the plant. Okay, the thickness. So the one that is responsible here, we can put, and that is question D. So that is the what? The cambium. Okay, so we can look at another function of the cambium is responsible in the of bringing about secondary growth in plants. So this brings us to the end of the questions for today. So please, if you've liked this video, kindly subscribe. Help us to reach out many learners out there. Okay, make sure that you invite some more so that within the time that is remaining, we can revise together. And like I always say, I wish you distinctions and see you in the next one.